Good morning, everyone. Thank you for your patience. I'd like to welcome you all to today's 10 a.m. briefing. My name is Matthew Wright. I'm the Deputy Director of the Science Press Package at AAAS. And we're welcoming you this morning to our 10 a.m. briefing, Infectious Diseases, Endangered Marine Creatures, Habitat, and World Food. Our speakers this morning are Drew Harvell, Professor of Ecology and Evolutionary Biology at Cornell University in Ithaca, New York. Carolyn Friedman, a Professor of Aquatic Fisheries Science at the University of Washington in Seattle. And Christina Miller, Head of Genetics and Genomics at the Pacific Biological Station for Fisheries and Oceans Canada in Nanaimo, British Columbia, Canada. We will first hear from our speakers, then we will open the floor to Q&A. A reminder that we will wrap at 45 minutes after the hour so we can prepare for our next briefing. At that time, our speakers will be available for follow-up across the hall in room 208. And now, our first speaker, Drew Harvell.
Yeah, stay there. Great. Thank you very much. Appreciate it. Thank you all for coming. We're really delighted with this. I reckon the coronavirus and things that are going on in the human world is on everyone's mind, as we're all especially impressed with how rapidly that emerged, the pace of its spread, and how massively it has changed our world already. Infectious outbreaks of ocean organisms are also fast and impressive in scale, but they're a lot harder to detect and track and see underneath the ocean. Widespread infectious disease outbreaks are fueled in the ocean by increasing human activities like land-based pollution and aquaculture, but especially a warming ocean and linkages with the changing conditions. Our session today will focus on new and ongoing outbreaks of ecologically and economically important organisms and their causes. So things like seagrass disease, starfish epidemics, oyster herpes, and salmon viruses. Every one of these are well-studied, globally spreading outbreaks that either diminish the health and functioning of our oceans or our capacity to feed the future. In every one of these cases, a warming ocean can accelerate the outbreak. So one example that I'll talk about very briefly, but I'll just mention here, is at the top of the food chain. This is a local Pacific-wide epidemic, and it's ongoing. It's imperiled starfish predator and was fueled by a warming ocean starting in 2013. This wonderful series of 2 pictures was taken 3 weeks apart at the start of that epidemic and shows the rapid death during this short period. The loss of this keystone is now disrupting the ecosystem, and we're involved in recovery efforts of that particular species while this epidemic's ongoing. So we got to keep in mind it's not all bad. These outbreaks provide new opportunities for innovation in fighting the age-old interaction between hosts and pathogens. And so all of us will talk about the increasing pace of new outbreaks, modes and rates of transmission, and some links with changes in ocean conditions. We'll showcase solutions and new transformative tools. For example, identification and quantification of the actual infectious agent in all of our talks is accomplished with new molecular aided disease dynamics. Dr. Christy Miller emphasizes that a warming climate spells disaster for salmon fighting disease, and Dr. Carolyn Friedman emphasizes the global intensification of an oyster herpes virus. In my talk, I'll describe the toll of an infectious disease of eelgrass is taking on an infectious disease of eelgrass is taking on the eelgrass of Puget Sound and the San Juan Islands, as well as more widely along our Pacific coast. We've found high and increasing levels of protozoan disease from 2013 and continuing. For both the eelgrass epidemic and for the starfish epidemic, these are coast-wide, so these are outbreaks that have spread from San Diego all the way to Alaska. I'll introduce a new artificial intelligence application that's allowing us to expand our scale as we can study from San Diego to Alaska. In our work, we use drones, satellite remote sensing, AI, and molecular diagnostics to aid surveillance of marine habitats. So, just so you know, after talking about all this death and destruction, I will end my talk by showing some results on how ecosystem services of habitats like seagrass beds have superpowers to fight pathogens and help perhaps sustain human health and biodiversity. So the commonality in all these outbreaks, the links with climate change, and the possibility for solutions triggered my writing this book this year, and I might say a little bit about that later. So that's all I have for now. Thank you.
Good morning, everyone. Whoops.
Yes.
No, this is actually my talk.
Oh, that's good. Let's see that. You want to brief us?
I'll now quickly go through 26 slides for you. Let's forget about my slides. So I'm here to talk with you about some of the work that we're doing on marine pathogens that cause catastrophic mortality in Pacific oysters. As Drew mentioned, a lot of these mortalities are associated with a warming ocean. My lab is focusing on the health of both wild and farmed shellfish, and in this case, Pacific oysters primarily. We've been working with a team with my laboratory, as well as colleagues, both nationally and internationally, to conduct research to help diagnose, characterize, and develop methods to control this lethal oyster disease. This is caused by the Oystriid herpes virus 1. We'll refer to it as OSHV1. There are also some other variants, and this is a suite of closely related herpes viruses that's causing mortality in oysters primarily. I know forefront in your mind, you're all thinking, oh no, should I eat oysters? Well, you cannot get herpes from eating these oysters. This is a virus that's very tenuously and distantly related from the herpes viruses that infect vertebrates, so don't worry. I work very closely with the shellfish aquaculture industry, located in Tomales Bay, California. This is in Northern California, where the Pacific Plate meets the North American Plate. And part of this region you may be familiar with, this is the San Andreas Fault. So a portion of the fault actually created Tomales Bay. And this is a very pristine location. It's very rural. There's a lot of farms, open grassland, forests, very few towns. It's a very nice location, and it's also one of the most productive shellfish bays in the state of California. So it's a very important aquaculture location. Unfortunately, beginning in 1992, the oyster growers started experiencing very rapid and intense mortality in their Pacific oyster seed. And this was happening during the summer months when the water was warmer. Colleen Burge, who's a faculty member at the University of Maryland, Baltimore County, who's a graduate student of mine, worked on this for her dissertation project and is still one of the leaders in the field investigating the herpes virus in oysters. I was going to show you a map that showed the global distribution of the herpes virus. It is found throughout, initially in France, throughout Europe, much of Asia, New Zealand, and Tomales Bay area in California. Unfortunately, after people started working with the virus and developing resistant stock, there was a change in 2008, and a more virulent form emerged. Seems to be rapidly changing, very adaptable, and is spread around the globe to a lot of locations where the original herpes virus was found, as well as to new locations. And when we polled the local shellfish industry here along the West Coast, the number one threat to their sustainability is the microvar. People feel it's not a question of if, it's a question of when it will arrive. So how is the shellfish industry supposed to adapt to a virus that's not yet here? How can they develop lines that are resistant to these strains when they don't know which strain is going to arrive? So some of the research that we've been doing is looking at trying to test different strains of virus from around the world, test different oyster lines in laboratory trials with these microvars in a strict quarantine facility, and then comparing these to how these lines perform in the field in Tomales Bay to see if there's a way to actually utilize the strain that is established or some of these strains in the lab to see if we can help them with their selective breeding programs so that they can survive these pathogens. And I'm going to talk more about that this afternoon. Thank you. Okay.
I'll flip forward
I'm so
quite
sorry
a few
about
slides.
that.
Close your eye.
Okay, so I'm going to be talking a little bit about
You want me to just flip through while you talk?
Yeah, you can get to my first slide. I only have 2 slides, so that's pretty easy. Salmon populations are in decline around the world. In Pacific Northwest, Chinook, sockeye, and coho salmon and steelhead trout have experienced precipitous declines over the past 30 years, and many populations are now threatened or endangered, and some are already extirpated. Climate warming is a major factor in this decline, with impacts that can directly cause stress in the salmon themselves, but also indirectly affect the prey availability and the predator distributions, as well as disrupting the natural balance between pathogens and their hosts. Our program applies a series of novel molecular and ecological approaches to understand the role of infectious disease and disease outbreaks in salmon declines, a difficult task when mortality is largely unobservable, especially in the marine environment. High-throughput pathogen monitoring tools have revealed the distribution and abundance of over 60 potential pathogens in salmon in over 28,000 wild, hatchery, and farm salmon. Models informed by a decade of infection data in juvenile Chinook salmon sampled during the early marine residence have revealed that temperature is the most significant driver of infection, whilst exposure to aquaculture, proximity to fresh water, age at ocean entry, hatchery or wild of origin, or latitude are associated with a smaller number of pathogens. Models are also being explored to resolve the pathogens most closely related to year class strength or cohort strength. And some are transmitted in freshwater, suggesting that the condition of fish coming out of freshwater is important to their marine survival, and some are transmitted in the marine environment. Importantly, models are also revealing that infection hotspots along the coast differ between the seasons. And they show that the Salish Sea within British Columbia shows an abnormally high rate of infection early after ocean entry in the spring and summer, while hotspots in the inlets off the west coast of Vancouver Island are stronger in the fall-winter period. Second slide. The mere presence of a pathogen does not mean that a fish is diseased. Disease requires that a pathogen is actually doing damage to its host. Disease is hard to study in wildlife when mortality is unobservable. Salmon in the ocean simply drop off from the water column, largely in the mouths of predators. We have shown that infection status is strongly correlated with risk of predation in rhinoceros auklets, which are birds feeding in the marine waters, and with freshwater bull trout in Fraser River. The most vulnerable fish are those that will be physiologically or behaviorally compromised by early stages of disease. Therefore, finding fish at late stages of disease, where it's easiest to document pathological changes, is very hard. As such, our program has also developed an innovative suite of host biomarker tools to resolve the earliest stages of disease, stress and disease development. One of the most impactful biomarker sets that we've developed to date is one that can identify the presence of a viral disease state using a non-lethal gill biopsy sample. This tool has identified that there are a number of fish showing signs of a viral disease with no detection of any of the viruses that we knew about, and has led to the discovery of over a dozen uncharacterized viruses and viral strains in salmon, both cultured and wild salmon in BC. We have shown infection of some of these viruses by applying molecular probes to visualize the viruses in the salmon tissue. For some, we've even localized the viruses in areas of tissue damage. Interestingly, for most of these viruses, the tissues infected are consistent with those of related viruses in other species, and with the known pathological effects in those species. One example is a newly characterized Pacific salmon nidovirus, which is evolutionarily related to the mammalian respiratory coronaviruses such as SARS, MERS, and COVID-19 currently causing outbreaks in China. In salmon, this virus also infects the respiratory system, and we find it infecting out-migrating smolts from hatcheries, salmon hatcheries in British Columbia, and it's in gill tissue. And it's increasing as juvenile fish are transitioning from freshwater to the marine environment. And we hypothesize that it may be associated with respiratory and/or asthmatic stress during out-migration. Another virus, named the salmon pescarinavirus, is distantly related to arenaviruses in mammals. The salmon arenavirus infects blood cells and macrophages, with areas of infection in the liver coincident with invasion of inflammatory cells, causing hepatitis. Hepatitis is the main pathological threat in the hemorrhagic diseases caused by mammalian arenaviruses, such as Lassa fever. In another study, we found a novel strain of a salmon hepatitis E virus, similar to hepatitis E viruses in humans. We found that infection alternates between organs, such as the liver, kidney, and spleen, to the brain. And this is what's shown here. This is actually showing the molecular probe to the Hep E virus in salmon in the brain, and that's all the red is the lighting up of the virus in the optic lobe of the brain. This is consistent with known disease impacts in humans and mice by similar hepatitis E viruses, where the virus can cause hepatitis when it infects the liver, but it can also move into the brain and cause encephalitis or brain swelling in others. Interestingly, this virus is statistically associated with encephalitis in salmon. The next generation of molecular tools named Salmon Fit chips were developed by my team to better understand the cumulative factors impacting salmon survival. The molecular tool applies a series of curated biomarker panels, similar to what we developed for viral disease, to identify specific stressors such as thermal, osmotic, and hypoxia stress, and a number of disease states. What's more, we've identified a molecular signature that's associated with imminent mortality, which recognizes fish that will die naturally within about 72 hours, regardless of the cause of death. All of this can be discerned with using the same non-lethal gill biopsy samples. We're now employing this technology to explore the complex synergies between stress and disease and to identify regions along the coast where salmon are the most compromised. The ultimate goal is not only to understand the combinations of stressors and diseases that have the largest impacts on salmon survival, but also to identify the stressors that can be mitigated. Reducing key freshwater stressors and disease in hatcheries and degraded freshwater habitats could increase chances of survival of out-migrating smolts. Mitigation of stressors and diseases with industrialization and aquaculture in critical early marine habitats for salmon will also increase their chances of survival. With overall survival rates plummeting to less than 1% in some stocks in some years, even a 2% rise in early marine survival could make the difference between sustained populations and extinction. Thank you.
Thank you to our speakers for those thought-provoking and concerning presentations. We would like to apologize to Professor Friedman for the mix-up with your slides and to everyone in the room. If you would like to get a hold of those slides, we will be able to email those to you. Please email media@AAAS.org and we will work that out. Again, apologies, but we would like to open the floor to questions. And please raise your hand, and we'd remind everyone to identify themselves by name and outlet, and we'll bring a microphone around to the room, starting with Robin in the front.
Thank you. Robin Williams from ABC in Australia. Drew, your news about seagrasses. Most people don't seem to know, which is a pity, that seagrasses absorb between 40 and 100 times the amount of carbon dioxide as do tropical rainforests. And yet they're being dredged all over the place. I wonder whether this new disease that you describe is depleting the populations drastically, or are they still surviving?
Thanks for that great question. Of course, the ecosystem services of eelgrass meadows are absolutely, and other seagrasses are essential worldwide. They provide habitat for many of our marine species. They provide homes for biodiversity, nursery areas, and as you point out, they're critical as a source of blue carbon and mitigating some of our carbon dioxide excesses on this globe, as well as the filtration services that I'm going to talk about in my talk. And sort of how we can be using nature's superpowers to combat some of these pathogens that are causing this trouble. We certainly don't want to be dredging and removing those habitats. Our work now is focused on understanding the role of the new disease. It's actually not a new disease. It's been around since the 1930s, but this new outbreak of this protozoan in diminishing the health and the coverage of our seagrass beds. And so I'll present some of that material in my talk this afternoon, but we are finding currently very significant reductions in densities of the eelgrass meadows in our local habitats in the San Juan Islands, along with very high levels of over 80% prevalence of this disease, which means 80% means 80 out of every 100 blades have a sign of the disease. So we think it is potentially a threat to the integrity of these beds in some locations. Yeah, good question, thanks.
Question in the back.
Hey, thanks. Brad Plumer from the New York Times. This question is for any of the panelists, but could you say a little more about the link between rising marine temperatures and increased rate of infectious diseases? Why does rising temperatures actually increase the disease spread?
Maybe I'll say something very general, and then they could give specific examples. I mean, we find that rising temperatures can both stress the host. So we've worked with corals for many years, and everybody probably in this room knows how corals bleach at warmer temperatures, but that also makes them more susceptible to picking up infections. At the same time, a lot of the infectious agents are more virulent or grow faster at warmer temperatures. And again, I'll say more in my talk, but these guys have incredible examples of it too.
As Drew mentioned, both the hosts and the pathogens are poikilotherms, meaning that their body temperature reflects that of the ambient environment. So it also means that their metabolic rate is driven by the ambient temperature. So when you have rising temperatures, if the temperatures fall outside, say, the optimum, perhaps for the host, they're going to be stressed. But if it's optimal for the pathogen, the replication rate and gene expression may change that allows them to become more infectious, more pathogenic, more virulent. And we see this a lot. You can have a change of 1 degree Celsius that can change a pathogen from being an organism from being pathogenic to non-pathogenic. An example is a Vibrio bacterium in France that infects abalone. It only infects the abalone when they're ripe, reproductively mature. And when the temperatures rise from 16 to 17 degrees, then the disease occurs and there's mass mortality. And this happens with a lot of diseases.
In salmon, we've developed models that have about 10 years of pathogen data in wild migrating juvenile salmon and explored the relationship with temperature variations along the coast, which there are quite a considerable amount of variation despite there's an overall trend of an increase in temperature. And what we find is that the vast majority of infective agents that infect salmon are positively influenced by temperature. And so to put this into context, a 1 standard deviation rise in temperature, which is about 2.7 degrees along the coast, will result on average on 25% higher prevalence of infection or risk of infection in salmon. If you look at the agents that are the most strongly connected with temperature, those that have been shown in the literature over and over again to be highly replicative in high temperatures, that jumps to 60%. So a 1 standard deviation rise in temperature, about 2.7 degrees, can increase the prevalence of some of those pathogens by 60% along the coast. And those kind of variations are occurring naturally every year, which means, I will show in my talk, in some years where temperatures are lower, we can see prevalences of some of these agents at 0 to 25%. And in other years, we can see 75 to 100% in those same populations. So that's how dramatically the thermal exposure can change the infection profiles and infective burdens in salmon.
Next question. In the front? If you would wait for the mic. Thanks.
Thank you. My name is Jacob Peterson. I'm a environmental journalism student at Western Washington University. I have a question for Friedman. Is the 1 degree Celsius increase that can make a pathogen, non-pathogenic organism pathogenic, is that on average increase?
So that's a good question. The temperature sort of a threshold for different pathogens can vary greatly, but when you're thinking about a 1 degree Celsius, it doesn't sound like a lot, but that is increasing the metabolic rate of the pathogens more than it sounds. So sometimes there's a threshold, sometimes it's a little more of a gradual increase for whether or not a disease is going to happen.
All right. Another question here.
Good morning. I'm Linda Mapes with the Seattle Times. So what do we do about it?
So in farmed populations, we have the opportunity that we don't have as much with wild populations in that we can direct selective breeding programs to actually select for animals that are more resistant to a particular threat or to a particular suite of threats. And that's been shown to be quite effective, particularly with marine invertebrates where the populations have a lot of genetic diversity inherent in the populations that allows them to change with a lot of these threats. And it's a balance between the reproductive rate and the rate of change that allows these to be successful or not.
Another question over here. Oh, I apologize. Sorry. Yes, of course.
Yep.
Well, the fortunate or unfortunate thing is that stressors can act both additively and synergistically. And when stressors are acting synergistically, which means that the presence of two or more stressors can actually create a much stronger effect, negative impact on the host or the organism. What that means is if we can even find a small number of stressors to mitigate, we can actually make a large difference in survival. My program's done a lot of work on temperature responses in salmon and thermal tolerances and the role of infection or temperature on infection. And what we find is that in salmon, as they're moving from either the marine environment into freshwater to come and spawn, or the freshwater environment to the marine environment to go to their natural feeding grounds, that the condition of the fish that are leaving those habitats, it makes a large difference in how well they're going to survive. So if you already have a fish, a smolt, that's coming out of the rivers that's already stressed, maybe it's not by temperature, maybe it's by disease or other factors, maybe they're coming out of hatcheries, already with stress. If you can mitigate those stressors and put out the healthiest possible fish to go into that marine environment, even when they're hit with additional stressors, they still will survive better. And we show the same thing going the other way with returning adults, that the condition of fish at the time that they're entering freshwater environments is highly predictive of how well they're going to survive the stressors, including temperature stress in those environments. So the key is to identify the stressors that we can do something about. There's a really great example for salmon in the U.S. actually, where they're controlling the amount of Serrata novashasta, which is a parasite that's a reportable parasite, causes devastating losses in juvenile salmon in fresh water. And they actually watch the levels of that parasite in the water, and when the levels of the parasite, which is highly responsive to temperature, when it gets too high, they actually open the dams and flush the system. And when they flush the system, they're not only reducing the temperatures and they're flushing down the parasite down the river, but they're also flushing down the alternate worm that is required to complete the parasite's life cycle. And so there are measures. It seems really drastic, and it seems like it's impossible to do in a wild context, but there are sometimes that things can be controlled using anthropogenic means.
One of the reasons for writing my book about ocean outbreaks was not just to draw attention to this increasing problem with climate change across the range of marine organisms, but to talk about solutions. And I think that there's some really innovative measures that we can approach if we start really thinking proactively. One of the ones that we've been focusing on, and I'll talk about in my talk, is that we found seagrass beds are natural decontaminators of pathogens that affect both humans and wildlife. So in working in Indonesian seagrass beds, we found that not only were the levels of human pathogenic bacteria reduced by 50% in water that went through those seagrass beds, but also the levels of coral disease were lower. And so this is, not a surprising notion that human health and biodiversity health can be linked, and there can be common solutions that are going to help our salmon as well and our oysters in some of these natural ecosystems. And I think it's a real important thing to start thinking about sort of these superpowers that some natural ecosystems have. There's quite a few other examples, but yeah.
Can I just mention one more thing? There's an example of an abalone disease in California, withering syndrome, that caused up to 95% mortality in black abalone. And a regulatory agency stopped fishing and protected the abalone populations, and some of the populations that experienced only 3 to 5% survivorship because of that wide genetic diversity, some of the animals are more resistant and they're actually rebounding. So by protecting some of these populations and allowing them to naturally develop resistance is another way to try and manage in disease, especially when you can't do anything about the temperature.
Similar to that in salmon, one of the things that's happened in British Columbia is a lot of the fisheries have moved to fisheries in river, from marine fisheries to freshwater fisheries. And what we know from a lot of study is that in terms of catch release fisheries, or fisheries where salmon may either escape or evade nets or be released from nets, that their ability to survive that capture experience is considerably reduced when you have high water temperatures. And so reducing those kinds of sport fisheries as well as net fisheries during periods when we have warm waters will make a huge difference in terms of the ability to salmon to actually make it to their spawning grounds and survive to spawn.
You got a question here, and then you'll be next.
Hi, so it's Kath O'Driscoll from Chemistry and Industry magazine, if this is working. My question relates really to, you were talking about the similarity of some of the salmon viruses to human disease. And I'm just wondering about the potential of some of these viruses for mutation and their transmission to other species, and whether that's also influenced by temperature rises, whether you're seeing any of this occurring, and whether it will exacerbate the problem, I suppose.
First of all, there's actually no examples of a virus being able to jump from a cold-blooded, vertebrate such as a salmon to a warm-blooded human. So we don't have a zoonotic risk in terms of that kind of transmission. Our temperature profiles are way too different, and viruses actually are, most of them, somewhat specific to their hosts. That being said, viruses can be exchanged between marine fish and salmon, and there's many, many examples of that. So that is very true, and temperature profiles may increase not only the virulence of some of those, but if they increase the virulence and increase the prevalence, then they may increase the amount of exchange and the number of species that may be vulnerable. So yes to being exchanged between a larger number of fish species, but no, not between a mammal and a fish.
All right. Next question here in front of the tripod.
Ann Thomas, freelance. Kind of getting back to your previous discussion about the various mitigation strategies, it strikes me that it's ultimately a race by how well you can improve management practices versus the rising temperatures and acidification. And I don't know that you can quantify the risks, but can you maybe each comment on how do you like our odds of being able to beat the warming?
Oh, that's a hard question. I don't like the way you asked that. Well, I think we can do better. Let's just say we're in our waters in Puget Sound, I feel like we're in a real vice grip between overpopulation increase, which has increased dramatically, and climate change warming increases. And so we really have to improve our game a lot. And I think there's a lot more proactively we can do. If you come to our session, we'll talk a lot about surveillance and diagnostics and improvements. So there's a lot of really good science that's being brought to the fore, and much more can be done. And I think we can focus a lot more on how to keep marine taxa healthy. And again, that's where some of our work with sort of natural pathogen fighting services in nature can be helping. If we're really convinced that eelgrass and seagrasses are important, then we really should be putting a lot more into conserving that kind of habitat, and everybody will be healthier. And there's some really exciting novel work. Sometimes we all spend a fair amount of time studying things that are sick and dying. But I think we can also turn our attention to ways to make things healthier. Dr. Friedman has a really cool example of a sort of a natural example of phage therapy coming to the rescue. And so and I'll let her comment on that, but there's things like that going on that we could be focusing on more. So I think there's a lot of solutions.
More questions?
But she might want to
Oh.
say something about her phage therapy.
It's not really my phage therapy. What Drew is referring to is, mentioned some emerging pathogens. Well, pathogens can have pathogens as well. So phages are viruses that infect bacteria. And the disease withering syndrome I was referring to, in about 2005, we started seeing what looked like a different new bacterium infecting the hosts. And when I did some electron microscopy, I discovered that these bacteria were actually filled with viruses, phages. And the phages basically hijack the bacterium and turn it into phage-producing factories, and it renders them non-pathogenic. And so, and it also is a defective phage, so they just kind of blow up like a balloon and just sit there producing phage, but not actually seeming to harming the host. So there are things that can change, and whether or not that's related to temperature, we don't really know. Temperature can have some different effects on changing distributions of both hosts and pathogens. So I think we're going to see a lot of changes in dynamics with the rising temperature, and some of these changes may be ones that are unexpected. If you have a rising temperature and a new species comes in, it can actually dilute the pathogen. Perhaps it's a host that doesn't become infected or doesn't cause any problems. So there's a lot of things that might change. Some changes, maybe a new host comes in and acts as a reservoir for a pathogen, providing more pathogen to susceptible hosts. So it's really hard to determine what's going to happen with rising temperature, and the pace, it really varies among the different hosts and pathogens because they all have different rates of change.
Question in the front.
Linda Mapes again from the Seattle Times, and this question is for Dr. Miller. Is net pen aquaculture contributing to disease risk in wild salmon?
Well, as I stated in the outset, the risk is not as great as changing climate. And that's what our models show. Our models show that the changing temperatures are impacting a much larger range of infective agents. That being said, there are a small number of pathogens that may be a risk of transmission between farmed and wild fish. And we are honing in on those and trying to understand of those which are the most important. And those might change between different species of Pacific salmon. So the things that are more risk for Chinook salmon may not be the same as those that are of most risk to sockeye salmon. And the other aspect of that risk really, and this is something we're dealing with in our models as well, depends on the life history of the fish. We know that the juvenile early marine rearing period is the most critical in terms of determining year class strength in salmon. So there's a lot of stressors, there's a lot of important aspects of life history throughout freshwater rearing and ocean rearing and coming back to return and spawn. But if you want to actually predict the variation in survival, it is happening in that early marine period. And there are different tacks that different stocks take. Some stocks, like sockeye salmon, they come out of the Fraser River and they immediately move north to Alaska. So their amount of exposure to farm salmon, they may swim by the areas of high density farms, but they may only be in that area for about a week and a half. Then you have West Coast Vancouver Island Chinook that come out of their freshwater rearing areas and they come into these inlets, and they can spend up to an entire year in that inlet before they begin to move offshore. And so our research is really moving towards the West Coast because we're interested in the potential for a greater impact of interaction with farms for stocks that actually spend more time in the vicinity of farms. And that is where we see hotspots of infection in the fall and winter period, and we're now trying to relate that to determine how much of that may be due to aquaculture.
Do you know which are the pathogens you're most concerned about?
Our models are just coming off, and I don't want to steal my postdocs' data because we haven't published them all. But I can tell you in generalities that pathogens that infect gill tissue are coming to the fore all the time. Pathogens that can cause anemia are also coming to the fore. We are finding a variety of parasites, viruses, and bacteria that are showing relationships with year class strength. The vast majority are actually parasites. Now, parasites are also the most common pathogens in salmon, and most of them are naturally occurring. Some are coming out of freshwater, some are coming out of the marine environment. We're honing in on some of these previously uncharacterized viruses that we've identified. One that I'm particularly interested in is this Pacific salmon nidovirus, which is the one that's a corona-like virus. Really because, and we have to do a lot more work and we need to do disease challenge studies, but the first step that we're taking to determine among these 12 or more viruses that we've uncovered is what can we find out from the tissue distributions of what tissues they're infecting, and looking for even minor levels of pathology in those tissues where the viruses are infecting? What can we find out about how similar the patterns are to what we know of viruses from the same family in other species? And so where we see those parallels, where we see that it's a virus from a family that causes respiratory diseases, and yes, it's infecting respiratory tissues, then we want to go, okay, well, that's one that we want to follow up on. And we know, as salmon are moving from freshwater to saltwater, that is the most critical time in their life cycle. It is so stressful. They have to undergo a complete cellular reconfiguration to be able to keep salt in freshwater and pump salt out in saltwater. And then the first month that they're in that environment is absolutely critical because they have to adapt during that period of time. It's also a period of time when they're hit with a whole bunch of different pathogens. And so they have to adapt and respond to those as well. So when those fish come out into habitats with higher densities of pathogens potentially, that's where we have to be concerned. And we do know that depending on the size of the fish, whether it's a hatchery fish or a wild fish, whether it's a fish that spends a limited amount of time in fresh water and moves out as a fry versus a fish that spends a lot of time in fresh water, all of those things are determinants of how much infection they'll have during that first month of ocean residence. And so it's complex, and those are the kinds of things that we're trying to unravel to understand how important is our farms relative to all of the other things that are going on.
Great. We have one more question in the back, and I think that might wrap us up for time.
Jeff Rice with the Encyclopedia of Puget Sound. You've talked about how viruses and pathogens are significant. Can you talk a little bit more about quantifying how significant they are? I mean, how many salmon are going to die, or how impactful is this going to be?
So in studying disease impacts in migratory species, we really have to go to using ecological approaches. We can use all of our molecular approaches to get the data, but then when it comes to quantifying those impacts, we need to use modeling. And we use the same kinds of models that have been used for years to look at relationships with El Nino and El Niña events and Pacific decadal oscillations. So we can use those same modeling approaches and determine the strengths of those relationships relative to other factors that we already know can impact salmon survival and returns. And so we're just at the early stages of comparing those. Right now we've been focusing on comparing the impacts of different infective agents. Now we're bringing in temperature and salinity, and what temperature and salinity can alone tell us about the predictions in variations in survival, and then we need to put it all together. So I can't tell you today, that there are, this percentage of fish are killed by X pathogen, but we will eventually get there with our models.
All right, thank you all. We are out of time, so we need to wrap up. But again, our speakers will be available. Thank you all to Dr. Harvell, Dr. Friedman, and Dr. Miller. They'll be available for a little while in room 208 for follow-up questions. But then, of course, if you'd like to see their session, that is at noon in room 6A. So I'd love to see you there as well. Thank you.
We survived it. So where do we